All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this topic, the plant growth and development, the subtopic, the plant growth regulators. Keep the NCRT textbook open with you. In plant growth regulators, the hormones which are chemicals, the diverse chemicals, simple molecule, non-nutrients, and organic substances, which are synthesized in one part of the plant body, whose effect away from place of its production in very low concentration, what do you call it is hormone. Production place at one place and site of action at other place in very low concentration, what do you call it is hormone. In literature, there are growth substances, plant hormone or phytohormone. Next, chemical nature of these hormones. Indole compound, that is auxins, and terpenes, gibberellic acid, and derivatives of purine adenine 6-purpuryl aminopurine, that is cytokinin, and derivatives of carotenoid abscisic acid, and other gaseous phytohormone, it is ethylene. And these phytohormones are broadly classified into two major categories based on the functions. One, it is the growth promoting substances like auxin, gibberellin, cytokinin. Growth promoting substances means involved in cell division, cell elongation, seed germination, flowering, fruiting, tropisms, and pattern formation. Very, very important word for NEET, pattern formation. Hormone is a great designer, architect of organism because the hormone decide when, how, where the organ has to develop. Such a wording, what do you call it is pattern formation. Next, the other plant hormone which having inhibiting properties, the what do you call growth inhibitor, like it is abscisic acid. In plant response to wounds and abiotic stresses due to abscisic acid, involved in abscission and dormancy, an inhibitor of metabolism. The other hormone which could fit either of the groups, promoter, inhibitor, but largely it is considered as inhibitor of the growth as far as ethylene concerned. Next, the discovery of each of the five phytohormone is accidental, that is serendipitous discovery. In case of auxins, Charles Darwin, his son Francis Darwin, while working on canary grass coleoptile, canary grass Phalaris canariensis, the part it is the coleoptile, that is the protective sheath of plumule, what they call it is coleoptile. And unilateral exposure to the light results in bending towards the light, what they call it as tropism. So response in direction of what it is the stimulus. So the relation exists between the growth and stimulus, direction and of stimulus, direction of response. What do you call it is tropism, phototropism. According to them, something synthesized in the tips, that is light perceiving, which is transmittable to downwards, causing the growth, but they could not notice anything. They not call it as auxin, something transmittable from apex to downward. If the coleoptile exposed to unilateral light, it is accumulated on shaded side. On shaded side, it leads to cell elongation bent towards light. Suppose the apex is capped, there is no bending because light perceiving part, something present inside the apex, is not going to receive what it is the sunlight. If the lower part of the coleoptile exposed to the light, it do not shows any bending. So something present in case of the tips transmittable in downward direction, that is light perceiving substance present in the tips. Later, F.W. Wendt, who isolated some substance from the tips of Avena sativa, that is oats coleoptile, who called it as auxins. Next, with regard to gibberellins, Korosova, who reported a disease in case of rice seedling caused by a fungus Gibberella fusiculae, and the disease it was Bakane or foolish seedling disease. The symptoms are the plant unusually grows tall, but there is no grains which are not filled. There is no use. Unusually grows tall. Grains are not filled. That's why it is a foolish seedling disease, Bakane disease. And sterile filtrates of the fungus, when applied to healthy plant, it shows the same symptom, that is the Bakane or foolish seedling symptom. Next. And cytokinin. Skoog and associate, they observed in case of tobacco plant, internodal tissue, that is medullary tissue, added with auxin and any of the following, that is extract of yeast, vascular extract from vascular tissue, DNA, coconut milk, all results in proliferation of the tissue. Next, Miller and others, they isolated the substance, they crystallized the substance, they called it as kinetin, the cytokinesis promoting substance. They crystallized it. Cytokinesis promoting substance, they called it as kinetin. Next, individual researchers responsible for abscisic acid, dormin, abscission 2, inhibitor B, all having the similar chemical properties, they called it as abscisic acid. Next, ethylene. Cousins responsible for the ethylene discovery. Ripe oranges, a volatile substance from ripe oranges which convert unripe banana into ripe banana. It is considered to be ethylene.